Vanakkam. Welcome to this course on classics in neuroscience. We have been looking at classic Nobel Prize winning works. In this series, we will be continuing our discussion of the classic Nobel Prize winning works of Edgar Adrian. In this class, we will look at how Edgar Adrian practically pioneered the recording from single neurons and isolation of single sensory neuron and isolating the single motor neuron, and how he was essentially the pioneer in recording from single sensory and motor neurons, and how that practically changed the field of neurophysiology in a very big way. So, clearly if you are studying nerves as a whole, you are studying multiple neurons, because there may be a bunch of neurons in one nerve bundle. So, for Adrian, an aim for him was to record from single neurons, single cells at a time. In this work, he was assisted by Yngve Zoeteman, a Swedish medical student. So, he made multiple attempts to isolate single neuronal fibers, but they were not very successful. Later, it was Zoeteman who thought of this technique of cutting muscle spindles that led to the isolation of single sensory fibers. So, Zoeteman's approach to cut muscle spindles in the muscles essentially isolated single sensory fibers and successfully recorded from the remaining axon, that also confirmed the well known and by now somewhat well accepted all or none behavior of the neurons. Remember, before this also there was some confirmation, but this was for the whole nerve bundle containing multiple neurons, but in this case the measurement is from a single axon, confirming the all or none law. Also, Adrian found that as he increased the load on the muscle, there was an increase in the firing rate of the neuron, that is force amplitude essentially was encoded in the rate, a very deep principle now understood as or called as rate coding or frequency encoding. This is now a well established principle of neuronal communication. Please do check my other videos in Neuroscience of Human Movements, in which I discuss this in considerable detail. That is, force amplitude was proportional to frequency of firing. Capital F is the force, small f subscript f is the frequency of firing. Here he showed that the firing rate essentially conveys the strength of the stimulus, and he found that neurons adapt to steady stimulus, suggesting that the sensory neurons respond to change in the load. This is absolutely critical. Respond to change in the load. So, that means after some time the firing rate stops, and then when you change the load, again the firing starts, suggesting that essentially this encoding is encoding of the change in the load. Now, very critical insight to make 100 years before. Now, this is well known and well accepted, but still this was groundbreaking at the time. Adrian and Zoeteman's findings were published in the Journal of Physiology in 1926. These findings are still recognized as a major milestone in the broad field of neurophysiology. This practically pioneered our understanding of how single neurons encodes sensory information. I already mentioned this, that this encoding is through frequency encoding of a change in the amplitude. That is what is happening. Note that depending on the type of muscle spindle, it can also be used to measure a sort of velocity, that is the rate at which the change happens, can also be measured, but that is a matter of detail I will not discuss in this video. I will leave you to check other videos or other resources for this purpose. Essentially, this groundbreaking work was perhaps the first one to record single sensory neurons and how they encode information or change in the stimulus. Adrian was also interested in studying single motor neurons. So, in 1928, Adrian and Bronk recorded from single motor neurons. They used the phrenic nerve that controls the diaphragm of anesthetized rabbits, and they dissected the nerve only until only a few axons, 2 or 3 axons remained without damaging them. They placed the electrode on the remaining axons and they observed bursts of impulses with each breath, suggesting that these neurons were essentially carrying the command for the lung muscles to contract. That is perhaps the first known recording from single motor neuron, but rather a few motor neurons, 1 or 2 or 3 motor neurons. This was the first known recording from motor neurons of live but anesthetized phrenic nerve of rabbits. Also, Adrian innovated by attaching a loudspeaker to one of their amplifiers. Recall that the amplifiers, the early amplifiers, were built in Adrian's lab by Alexander Forbes in the 1920s, early 1920s, but this was 1927-28. By the time Edgar Adrian must have already had multi-stage amplifiers, so he attached a loudspeaker to one of their amplifiers, and this enabled Adrian and his colleagues to hear a clicking noise every time a neuron fired. This method later became a standard method of recording. Listening to nerves clicking became a routine approach in Adrian's lab and was later adopted by many labs worldwide, that a nerve is active when you hear this sound in the speaker. It is very simple, very easy to identify. You stimulate and then you see whether you are hearing crick, 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 like that. Of course, you can also count the number of pulses and find the frequency, but that is another step forward, but at least you can hear the sound. With this, we come to the end of this video. In this video, we looked at Adrian's attempt to record from single neurons, in particular how his collaboration with Zoetemann led to isolation of the muscle spindle, and how it led to the groundbreaking finding of how force changes or changes in the amplitude of the load essentially were encoded in the frequency of firing or rate coding of the sensory neurons, that is the muscle spindle, and also how he used the anesthetized rabbit as a model system for studying the motor neurons by recording from their lung phrenic nerve that supported breathing. With this, we end this video. Thank you for your attention.